All right, so as everybody's getting seated, I'll go ahead and do an introduction to the second session of the first ever and hopefully annual IPSS joint collaboration with Plant Pathology's 3-minute thesis competition. And as I mentioned this morning, we have changed our 3MT to fit our departments, and we are including students, staff, and faculty in this presentation. So we're glad that you all are here, and we're glad that we had a lot of faculty and a lot of students, and hopefully next time we'll be advertising more so we can include more people in our departments. So I guess a real quick rundown of the rules. Speakers have 3 minutes. Once the 3 minutes passes, they will have a slide that looks something similar to this, and they are to stop talking. And in the rules, they're also disqualified from winning. So we're glad you're here, and without further ado, I'll let our first speaker, Stu, come up and take over. And for the speakers, if you're going to walk away from the podium, please take this mic with you, and I will stand up whenever there's 30 seconds left. This thing on? Testing, testing. There it is.
Cool. So in modern agriculture, there's an increasing demand for safe and specific alternatives to traditional small molecule pesticides. Perhaps one of the most promising of these is known as RNA interference. In RNA interference, a pest species of some kind is exposed to double-stranded RNA. A gene that corresponds to that sequence of double-stranded RNA is reduced, and if an essential gene is targeted, the pest organism will die. However, there are some complications associated with this technology, namely that double-stranded RNA is not very persistent in soils and outside of the organism, and a number of insect species are actually recalcitrant to the uptake of double-stranded RNA. There are some ways that we can overcome these shortfalls in RNAi, one of which is complexing double-stranded RNA with a nanoparticle carrier. However, this introduces further changes to our traditional understanding of double-stranded RNA. It changes the way that the double-stranded RNA may either be taken up by the organism or degraded in the soil environment, which is what my project is going to focus upon. So we've already seen that in some of our results, where we see that mutant organisms exposed to double-stranded RNA nanocomposites that are normally resistant to the uptake of double-stranded RNA show severe gene knockdown when exposed to these particles. Our future work is going to focus upon the degradation of these particles in natural soil environments. We will be labeling the different components of these particles with tracers and monitoring the kinetics of dissolution depending on different environmental factors. The overall goal of this is to achieve a safe, sustainable nanocomposite that's available and improves and allows the production of safe pesticides into the future. Thank you.
So just like the last session, we'll have a 2-minute break in between speakers that were our judges can finish scoring, since they are judging one talk and there's only 3 minutes to judge during the talk. So we'll wait another minute before our next speaker.
Right. Shut down the screen.
Yep.
My best timing is 3 seconds after.
I mean, they put it for
Hi, RNA viruses are widespread pathogens, not only in human animals, but also in plants. During the virus infections, it is a battlefield inside the plant body. Viruses subvert host membranes to build replication organelles and co-opt tons of host factors to facilitate its own replication. In the meanwhile, plants also have been evolved to combat virus with different types of antiviral responses. In order to develop a solid and a sustainable antiviral strategy, we can learn how plants suppress viruses from their long-term arms race. How do plants suppress virus infections? One way to fight against viruses is using the endogenous restriction factors in the plants. In my study, I have identified a RNA helicase RH30 as a restriction factor that inhibits the accumulation of 5 RNA viruses in plants. To understand the mechanism of this inhibitory effect, I have tested the subcellular localization of RH30 and during the virus infection and the protein-protein and protein-RNA interactions. Here's what I found. So as you can see on the figure, after the entry of RNA viruses, the viral replication proteins are translated and then recruit the viral RNA template as a template to initiate the viral replication. During this recruitment, RH30 steps in and targets the viral protein RNA complex. And as like its cellular function that catalyzes the ribonucleoprotein complex rearrangement, RH30 remodels the crucial secondary structure on the viral RNA template, which prevents the viral RNA template being recognized by its own replication protein. As a result, the viral replication proteins are no longer be able to recognize and recruit its own viral RNA as a template to initiate the replication. In this research, RH30 shows a novel antiviral strategy against broad-spectrum RNA viruses in plants. And more importantly, through studying the interaction between host and plants, we might learn more, gain more knowledge to develop a solid and sustainable antiviral strategy along with the latest genetic engineering. And one day, we might be able to turn the table and eliminate viruses from Earth. Thank you.
Well, the last session we had somebody tell a joke. Anybody know a joke? I know our judges don't like any joke? No? I guess that works. Looks like our judges are good, so.
Appreciate the second chance. Thank you so much for your attention. Many of us working very hard here to contribute to the sustainability of agriculture development. So how many of you just use computers without any field experiment instrument and laboratory studies? None, except for my lab. My lab uses ecosystem modeling approach by integrating multiple databases to identify climate-smart agriculture impacts on crop growth and soil biogeochemical cycles. Those practices, including cover crop, conservational tillage, and biochar, have been investigated in many long-term and short-term studies, including field experiments, remote sensing, and sensors. we synthesize multi-database at plot and field level to watershed and regional scales that help us to understand how the whole agri-system model, agri-system in response to those practices. So we also need to improve the model to incorporate those influencing mechanisms. We also need to develop a regional database to drive the model. Then, the final step, through data model integration, we can predict the regional dynamics of crop yield, greenhouse gas emission, nutrient leaching, soil carbon and nitrogen, as influenced by diverse management practices. Such information of regional assessment to benefit us, the development of climate change mitigation strategies, and the development of decision support tools for stakeholders and policymakers. That's a long way. And obviously, our research involves interdisciplinary studies in crop and soil science, atmospheric science, environmental science, computer science, and remote sensing. So we do need your collaboration and support to make it. Thank you.
So just a reminder for everybody, we are doing a People's Choice Award for your favorite faculty and your favorite student. So make sure before you leave the second session that you vote for who you thought did the best in the first and second sessions. Since the faculty are not being officially judged, we'll go ahead and proceed with our next speaker, Austin.
A short time ago, in fields not far away, there was a weed, mare's tail, and it has been found throughout the United States to be glyphosate resistant. Researchers, extension agents, growers, and quite a large number of people find this lack of control disturbing. And in addition to that, it has been found in Kentucky to emerge, even though it's a summer annual, to emerge anywhere from early March to early November. And so with that type of opposition, force choking at us grain crops out of the picture, we need some sort of really important control. Enter cover crops. Cover crops are not a relatively new idea, but researchers are currently looking at them with herbicides for best management control of really troublesome weeds such as mare's tail. And my project is focusing on that very issue. I have a fallow field in Winchester, Kentucky that is looking at mare's tail control prior to soybeans. And we are, as you can see on the left, you see the mare's tail right here front and center, but you see that greenish hue in the background? Those are my plots for this coming summer. So we have a lot to work with. Cover crops have been proven by previous researchers to control weeds because they are able, especially like mare's tail, because they compete for light, for nutrients, and especially for seeds like mare's tail that emerge mainly from the soil surface. It's very imperative to get that type of coverage. But I'm also looking with herbicides. I have a 3-factor program that is fully factorial, looking at a fall-applied herbicide called Sharpen, a spring-applied growth regulator, either 2,4-D or dicamba, and then cereal rye cover crops. Though I am still working through some of the interesting statistical analyses of my first year of data, I have found a couple of interesting notes. First of all, where we have cover crops just by itself, it has significantly reduced the mare's tail population. And as far as soybean yield data, we didn't have any significance from this prior yield. So even though I have a lot more analyses to do, along with another whole year of data, will hopefully be able to shed some more light onto exactly how mare's tail can be inhibited by herbicides and cover crops. Hopefully, we can stop the mare's tail empire from striking back. Thank you, and may the force be with you.
Also, keep in mind, this is the first time we're hosting this event. The GSA and Graduate Student Association of IPSS teamed up with the Graduate Student College Department, and we tried to put on an event that would be, I guess, short, fun, a lot of people can participate in. So if you have any suggestions, reach out to the GSA or either one of those for the future. If you like this event and anything about it, change it. We're ready whenever you want.
Well, hello. Imagine that you are a livestock, a forage producer for these 3 minutes, and you're trying to raise this one here as the better you can. You put her in a grass pasture as usual, and you and you let her graze. But by doing a little research, you find out that cattle loves the red clover, and you are determined to interseed the clover in the grass pasture. By doing that, you find out that you end up increasing your soil fertility, your forage quality and yield, and also you find out that your cows are very happy. And by doing that, actually, you create a problem, although it works perfectly, because the weeds start to come out. And when you go to your herbicide catalog to try to control those weeds, you don't find any option that will not harm the clover you've just seeded. This is the main issue Kentucky forage growers face when trying to work with a clover grass mixed pasture, the lack of herbicide options. Yeah, to address this issue, the University of Kentucky Forage Breeding Program developed a new variety called UK 2014. UK 2014 has displayed an increased tolerance to the herbicide 2,4-D, which is one of the cheapest and most available herbicide options in the market. However, the increased tolerance in UK 2014 hasn't fully been described, and most of the biological processes are not clear yet why this increased tolerance happens. Our aim in this work is to address this question, and in the end, to deliver a novel variety that's flexible to growers and allow them to gain the benefits of the clover and still maintain an herbicide in their weed management program. Our preliminary results demonstrate that UK 2014 has similar yield to another variety commonly grown in Kentucky. Moreover, UK 2014 has less injury from the foliar applications of the 2,4-D herbicide and also has a better regrowth after harvest following the treatments. Herbicide metabolism assays also demonstrates us this new line has the potential to metabolize the herbicide molecule and also has a reduced translocation in plant. These results put together demonstrates us the great potential of this line to be adopted in a grass clover mixed pasture. And it will deliver a novel and flexible option to forage growers. In summary, we know the capability of this new line, and we hope that it makes the growers as happy as this cow here. Thank you.
If you have seen the schedule, unfortunately, Dr. Vincelli couldn't make it today, so we're all going to have to skip him, and we'll be continuing with Rebecca.
Sorry, one more.
Growing up in Ohio, you are infused with a pride in Lake Erie from birth. Ohioans love to call it Ohio's Great Lake, and we have a great deal of pride. But it becomes harder and harder every year to have pride in Lake Erie when NASA releases a photo like the one at the top of the screen. All of that green on the left-hand side of the photo is algae. Lake Erie, like many freshwater ecosystems around the world, are phosphorus-limited systems. So when phosphorus is added to Lake Erie through nutrient additions from farm runoff, biomass production can begin, and in the case of Lake Erie and other systems around the world, you get algal blooms like that. See, the application of inorganic fertilizers and manures to our farm fields creates a readily available source of nutrients for our crops so they can grow. But it also creates a readily available source of nutrients that can run off our fields during rain events. They can erode with the soil, and they can even leach out through the tile drains and eventually work their way into our water systems. So what do? Do we go back to farming the way my great-grandfather did on his farm over 60 years ago, without inorganic fertilizers and working his farm field with horses? Well, it's not quite that simple, because the application of fertilizers and manures has led to a buildup of legacy phosphorus in the soil. So we still have phosphorus there, and it's not quite in the right form that the plants can use. So what are we going to do about it? Well, I suggest that the way we get to the root of the solution is doing just that. We look at the roots of the plants we're growing. Crops were previously grown in societies without using fertilizers or these nutrient additions, and the crops still had ways to grow with the nutrients they needed. But breeders have only looked at the above ground traits of these crops. They haven't looked at the roots. So we have selected a panel of both historic and modern varieties of winter wheat, a common cover crop in the United States and a crop my great-grandfather grew on his farm. And we're going to grow these winter wheat varieties in a clear media so that we can photograph their roots and then measure them for different architectural traits such as length, surface area, and even branching angle to try to figure out what has occurred in breeding events that may make our crops need that inorganic source of nutrients. I'll also be measuring the production of organic acids by these roots that help to solubilize forms of phosphorus that may not be readily available to the plant. In this way, we can start answering the problems of solving that ugly algal bloom on Lake Erie just by growing different crops. We won't be adding any more fertilizers. We'll be drawing down on that legacy phosphorus, but we also won't have to go back to working our farm fields with horses like my great-grandfather did. Thank you.
Hello. So one of the reasons why we wanted to do this event was also to get everybody out of their offices and labs in the field and get in one room, and we've actually filled it up pretty good, so thank you for being here. So in the meantime, when judges have the 2 minutes in between speakers, feel free to maybe not get up and move around, but talk around to the people around you to see what's going on in their days. So we'll give another 30 seconds or so before our next speaker, Kai.
So what do you need to do?
Cookiestar is one of the most important methods for accessing the expression panel of target genes. I believe the majority of individuals in the audience could utilize Cookiestar in their research. We all know that successful cookies are rely on the use of high-quality primers. I want to propose a question. How long will it take you to design a Cookiestar primer? 1 minute? 10 minutes? For some individuals who are not expert at molecular biology, it could take days. To save valuable research time, researchers now can access a Cookiestar primer database, which can design a Cookiestar primer for you in less than 10 seconds. Now you are probably thinking to yourself, oh great, another form of software I must learn to use. Actually, it's extremely easy to use this database. Simply show on this slide to open your designed primers. You just need to access the website of this database and follow 3 steps. First, select organism from the drop-down selection menu. For instance, you can easily find your Embryogenesis from the plant classification. Then you will research primers based on the target gene using the associated gene ID, gene name, or just the gene description. Once the gene information is entered, press the search button and the database will generate available primers for your target gene. You can only select to only display the best primer pair. Details regarding the primers, including the GC content, the melting point, and the applicant sequence are also provided. For more information regarding this database, please refer to our paper. Don't hesitate to contact us for providing the feedback and comments on this database. As with any scientific endeavor, this database is consequently upgrading, and any comment will help us to build a better database, which will be extremely helpful to everyone's research. Thank you.
Yeah. It would be nice to have. Yeah, that's right. I'm gonna do.
You're all in favor of soil health, right? Right. What's the alternative? Unhealthy soil? Well, that's just crazy. But what is soil health? It's the capacity of a soil to function as a dynamic ecosystem to sustain plant and animal life. We know it when we see it. At least we think we know what it is. A healthy soil has 3 components to it, and you have to have all 3 for a healthy soil. Optimal physical properties, optimal chemical properties, and optimal biological properties. When they intersect, you have optimal soil health. Now, think about your own soil health. If you want to improve your health, your personal health, if you want to improve that, you've got to work at it. And the goal of soil health is to move where you are at one level of soil health and improve the area of soil for those physical, chemical, and biological properties so that more of that soil is in an optimal condition, and therefore you've expanded the area that involves or represents all the soil health. If you can change something, you can measure it. So how do you measure soil health? Not all of these properties change at the same rate. For example, the physical properties don't seem to change very much at all, but we can measure them. We can measure bulk density, or penetration resistance, or aggregate stability or aggregate size to tell us that we've made an improvement to soil health. Chemical properties change a little bit more quickly, but we can still look at that change in terms of a change in the pH, or a change in the nutrient holding capacity, or a change in something like total nitrogen or total carbon. But look at the biological properties. They change a lot. They're very good short-term indicators of whether a soil is getting healthier or not getting healthy at all. And that's where our lab comes in. We like these short-term changes. We like to measure the biology, the respiration, nutrient management, labile carbon. Think of us as an agricultural HMO. We provide recommendations to make soil health better in terms of soil management, and we provide the measurements that allow you to identify that you in fact have improved soil health and are therefore increasing your soil value. Thank you.
I want you all to think about what you've eaten this week. Do you think you've eaten more pork, more beef, or more chicken? If you're like most Americans, the overwhelming answer is chicken. Americans eat over 90 pounds of chicken per year, and that's way more than pork or beef. Poultry consumption has been on the rise, which has led to an increase in poultry production facilities, not only in Kentucky, but also in the surrounding states. Kentucky's #1 commodity is poultry, and there are over 3,000 poultry houses in Kentucky alone. With that many poultry houses, as you can imagine, there's a lot of waste that comes from there. This waste is known as poultry litter. Poultry litter is the combination of the bedding used in the poultry houses and feces. Sounds pretty great, right? Well, it is for crop producers. Many farmers choose to use poultry litter as a fertilizer on their farm. It can be a very beneficial and a very economical choice for them, depending on how close they are to the production facilities, depending on the cropping system that they have, and then also on the nutrient availability in their soil. One important process that occurs when you apply poultry litter is nitrogen mineralization. This is the process where microbes break down organic nitrogen into a plant available form. This is very important when applying poultry litter because it determines when and how much nitrogen will be available for the crop. There are many different factors that impact this biological process. The physical and chemical composition of the poultry litter and the soil impacts at what rate this process takes place. I'll be studying some of the factors that influence this rate. I'll be looking at the different bedding materials that are used in poultry production facilities. Those are primarily wheat straw, wood shavings, rice hulls, and sawdust. Each of these bedding types has a different physical and chemical composition, which in turn impacts the composition of the poultry litter. And I hope to be able to tell how big of an impact this makes on the nitrogen mineralization rate, and if it does have an impact, what is it? The other thing that I'll be looking at is another factor that influences the nitrogen mineralization rate, and that's temperature. I'll be looking at how in Kentucky, when producers apply in the fall versus in the spring, how this impacts when and how much nitrogen will be available to them and for their crop in the spring. Overall, the goal is to help Kentucky crop producers use poultry litter more efficiently and more economically. Thank you.
And this is for I want to use these 3 minutes to your attention to give you a brief description of what my program is about and what I've been working on. My focus is in understanding how crops interact with environment and with management options and how this affects development, growth, partitioning, and finally yield. And I find this fascinating because in fact all crops are governed by similar sets of rules of how they respond to environment, which is what makes it possible to build crop models. And I want you to think of crop models as a puzzle, where each of these pieces is our understanding of an individual process. With research trials, we can only look at one of these processes at a time or answer a limited amount of questions. We often have control conditions where we only modify one factor at a time. But with a crop model, we can put all these pieces together and look at the whole picture with still enough detail that we can get quantitative answers. And I think this approach and these tools can be very important for answering challenges in agriculture. Because if we want to put more weight on the supply side of this balance, we need to rely on all available tools. Not all the best breeding efforts or the best technology will perform best in all conditions. But we need to tailor these recommendations for each environment, and there's even going to be year-to-year variability. So we might need to diversify our management. And answering to this complex interaction will involve a realistic amount of data, and I say massive fieldwork, and my students might think, but we still did massive experiments. But that's still looking at a very tiny fraction of this complexity. And I want to end with an example of the potential of looking at this genotype by environment management interaction with some results from last year. In this graph, you can see the bars in dark blue show yield for soybean from early to late maturities, and in light blue is soybean yield under irrigated conditions. So we found that if a producer switched to an irrigated system, or if there's a soil with more water available, they need to be changing their maturity choices. And most importantly, and we would not see this if we were not to use a modeling tool. If we look at water use efficiency, even though some genotypes have similar yield, these management options are still having a high impact on water use efficiency. Thank you.
Is it possible that the answer to the world's food shortage problem can be solved with something so small as a single microorganism? Something can be grown in a paper dish? Food security is a major concern for today's society, and it is estimated that we may not be able to produce enough food to support the next generation. One way that we can increase food production is through the use of herbicides to eliminate weeds. Unwanted plants growing alongside the crops are known as weeds. Why are they a problem? They have fierce competitors absorbing water and the nutrients with little remaining for the main crops. They also grow really fast, creating shade around the plant, prohibiting their ability to photosynthesize. Herbicides are an important tool in modern agriculture. However, to eliminate weeds, weeds often develop resistance to herbicides. Therefore, we are searching for novel ways to attack these undesirable plants, creating our food supply. Creating our food supply. Creating our food supply. For my dissertation research, I am searching for novel ways to attack these undesirable plants. The microorganisms living within a plant. These microbes, plants have evolved a complex relationship with microorganisms. Some of these relationships can be classed as mutualistic, where both organisms are supporting or helping each other. Other relationship could be a parasitic, which is my main interest in this research. In order to identify the microorganisms that are plant parasites, we isolate the bacteria from a plant, and then we culture them in our lab. They were then used to inoculate the seed, and the effect of the inoculation were measured at a specific time interval. I determined that a particular bacterial strain inhibits the plant growth, specifically inhibiting the production of cellulose, a vital substance that is important for cell wall structure. Plants without cellulose will die. However, identifying the bacterial strain is just the first step. I am now working with the researchers at the School of Pharmacy in order to identify what substances are being produced by this bacterial strain. If we could figure this out, we are in our way to produce a new herbicide. It is my hope that this finding will lead to the production of a novel herbicide. Herbicides produced by microorganisms could be highly selective and less likely to have off-target effects, and they will be more environmentally friendly to support our food security. Thank you.
So my lab question is, how do plants control fertilization and seed development at molecular and cellular levels? So we are using genetics, genomics, and live cell imaging technology to find out those processes. And this is the confocal image showing the egg cell inside the seed. So the nucleus is yellow, egg cell nucleus is yellow, and the egg cell actin cables, it's one of the cytoskeleton structures in the egg cell, and then the seed cell walls and the plastids are in magenta. So we found that these actin cables are playing an important role for sperm nuclear migration to the egg cell nucleus for fertilization. Unfortunately, I cannot show any video clips today, unlike Dave. But I hope your imagination, I'm gonna rely on your imagination to picture this beautiful dynamic egg actin cable movement inside the egg cell. So actually these actin cables are generated at the plasma membrane periphery of the egg cell. And then these generated actin cables are moving towards the center of the cell where egg cell nucleus is located. Once sperm cell nucleus enters, then somehow, we don't know yet, somehow sperm cell nucleus is recognized by the egg cell actin cables, captured and transferred to the egg cell nucleus for fertilization. We know actin cables are important. We know there's a beautiful dynamic movement of actin cables inside the egg cell, but we have no idea how this movement is controlled and what factors involved in the recognition of sperm nucleus for fertilization. So now we are in the process of identifying those factors using Arabidopsis thaliana as a model. Interestingly, this usage of actin cables for fertilization is very unique to flowering plants. In ferns, liverworts, and animals, they don't use actin cables for fertilization. They use microtubules. More importantly, this unique usage of actin cables is happening during seed development as well. So our preliminary data suggests that manipulations of this actin cable dynamics in a seed can alter the final seed size. Therefore, we are developing new project to understand how this actin dynamics can impact on the final seed yield, which is important for agriculture. Thank you.
So ascorbic acid, normally known as the vitamin C, is a very important antioxidant for human health. So, we human mainly obtained from our diet, like fruit and fresh vegetables. However, in plants, the plant can make its own. The synthesis pathway. The synthesis pathway start with the conversion from mannose 1-phosphate to GDP mannose. Ascorbic acid are suggested to minimize the generation of reactive oxygen species, also known as ROS, by the catalyzing of peroxidase. Ascorbic acid can reduce the hydrogen peroxide into water. In human beings, ROS can cause aging and cancer, while in plants, ROS also regulates the immune response to pathogens, as well as the immunity our lab has been studying, which is called systemic acquired resistance. Maybe many of you already heard from the last section. So this is the general picture what ICR looks like. So it's there's the infection in one part of plants. A certain kind of chemical are produced. Some of them can travel through the plant to the other part, which result SAR. And these immunized plants are ready to against the future infections by many pathogens. So our focus is on this signal pathway. So we are studying how and which chemical are produced in this pathway, and how this chemical were transported through the plant, and how does the other part of plant receive this chemical signals. So one of the chemical signals is ROS, as I have mentioned before, and the implant ROS level is in a very specific range, and the SAR can be induced. So my project is try to understand whether the level changes to ascorbic acid also can affect SAR, and how does ascorbic acid affect SAR. So for doing that, I'm studying into this VTC1 protein. It's the rate limiting enzyme in ascorbic acid biosynthesis. So in Arabidopsis, a mutation which loses the function of VTC1 results a 17% decrease of ascorbic
acid levels and cause the high level of loss and also high basal resistance to bacterial pathogens, Pseudomonas syringe. So I'm going to look at the genetic mutation of VTC1, and I'm also looking at its biochemical activities, and try to study how does VTC1 interact with other known compounds in ICR signaling, and how does ascorbic acid affect ICR? Thank you.
Today, I'm going to share a story. It is a story about the present and our coming future. It is the story of the seeds. Did you know that 70% of our diet consists of seeds? The increasing temperature and growing population around the world has caused the scientists and researchers a real problem to produce more seeds. Being molecular biologist, my research focuses on gene regulation on growing more seeds, what genes and proteins are involved in it. Our lab basically is interested in studying the proteins and genes which helps in embryogenesis, that is the process either by seed or artificial process called somatic embryogenesis. Previously, our lab has found an important gene, AGL15, which is considered as a master regulator in somatic embryogenesis. It might be surprising to know that how people interact in society to do some work. Similarly, the genes also interact with each other to perform some function. For example, AGL15, it interacts with some genes by inducing their function, whereas repress, that helps in embryogenesis. And my research focus is based on another gene called LBD40, also called LAB40, which is seed-specific and previously found interacting with AGL15, which means this makes an interesting topic for us to understand how seed embryogenesis takes place. So my approach will be on working basically with the Arabidopsis plant, which is model plant, and embryo culture tissues, which we made in the lab, and using CHIP and transcriptome analysis, I will start my work. CHIP is chromatin immunoprecipitation. In this process, we use chemical to fix the proteins and study protein-protein interaction by eluting in a blood. Whereas the transcriptome analysis means studying the genes which they are upregulated or low regulated during embryogenesis. And combining this process, we will come up to a working model to see what happens if there is no LAB40 gene in plants or when there is overexpression of LAB40 genes. And as you see, there will be a complex web where there are some genes which are directly interacting with AGL15 or which are indirectly interacting, which helps to produce embryos. So understanding this scenario will definitely understand us to know the embryogenesis in Arabidopsis plants. Then therefore we can use it on other crops which help us to grow more seeds and will feed the future. Thank you.
So currently went in the first session, he took over for another speaker that couldn't make it. So this concludes the second session, but before you get up, make sure that you visit the back of the room and vote for your favorite speaker. Vote for a faculty member and a student speaker, please, before you leave, and come back in about 30 minutes for us to share the awards. Let's thank our speakers. Also, there will be a social event at, where's Kirtley? Henry Clay at 7 o'clock tonight. 7:30. 7:30, Henry Clay, social event. Is somebody in Dr. Perry's lab? Or running by Dr. Perry's lab? Sanjay? Sanjay, you still here?